So let's dive into detailing enzyme active sites for the four enzymes that we're going to study. They're all hydrolysis enzymes, so they are using water in some way or shape or form to either break linkages that we have between monomers, or with carbonic anhydrase, we're just going to see the involvement of water. So we're going to treat all of these the same, all of these little videos the same. We're going to give a little bit of background as to what this enzyme is, how it works, any important key structural details, and then we're going to look at the uncatalyzed reaction to understand kind of what we saw when we did those reaction mechanisms, and then we'll map that onto what we see in the enzyme active site. All right, so here's what we have for RNase A. So RNases are enzymes that degrade RNA. They're going to hydrolyze or break the linkage that we have in those nucleotides, so that break that phosphodiester bond. We have them because RNAs are not normal sort of long-term in our bodies. Whether they're messenger RNAs that are meant to be short-lived or viruses that are foreign, your body knows that RNA is not supposed to be something that sticks around for a long time. So it has mechanisms to get rid of RNA that's sticking around for too long. RNase A is a specific kind of RNase. It's actually produced by your pancreas and secreted into the GI tract. So it's kind of one of those things that sort of helps make sure whether they're viruses that kind of come into your GI tract. Remember, your GI tract from start to end is really the outside environment that kind of goes through you, and your intestinal tract sort of pulls out information, nutrients, all kinds of things from that outside environment. So lots of things to make sure that we break down those nutrients that we have in there so that we get them out, or in this case, make sure that if there's foreign things that are there, that maybe they're not doing some bad things. Okay, so you do not need to know sort of structural details, but this is actually a fairly small protein, 124 amino acids. The catalytically active residues that we're going to pay attention to are 2 histidine residues. Now, the neat thing with histidine, I don't think I've mentioned this before, is that histidine has pKa's that are near neutrality. So unlike the carboxylic acid functional groups that we see with aspartic acid and glutamic acid, or the other protonated amines that we see with arginine and lysine, which have pKa's 9 to 10, the carboxylic acids 3 to 5, histidine has a pKa closer to neutrality, just a little bit above 6. And so that's going to allow it to serve as both general acid and general base catalysts. So just zooming in here a little bit to the enzyme active site, here we can see in red, we've got our substrate, a little piece of RNA that's there. We can see this enzyme in particular has a couple of disulfide linkages that sort of helps it kind of stay together. You may or may not remember, we actually looked at RNase A when we talked about protein folding and misfolding, and the importance is of having properly connected disulfide linkages to dictate who's going to be next to who and form a properly folded tertiary structure. So here we can see that we've got our histidine residues here that are going to be properly doing their acid-base chemistry. Okay, so let's take a look at the uncatalyzed reaction. So a couple of slides here that are going to build on one another. As we look at our uncatalyzed reaction, and when we did these sort of as the reaction mechanisms that we turned in, we didn't necessarily look at a phosphodiester bond in the context of a nucleic acid. We just sort of looked at that phosphodiester bond. So just making sure to highlight, right, we've got our phosphodiester bond here. And this is the bond that we're going to be looking at breaking, this bond right here. So some themes that we have seen when we look at reaction mechanisms. If we want to have a bond break, that means it's leaving with its electrons. And so we can actually facilitate that leaving group leaving by protonating it. So that's something that we're going to see. Okay, nucleophiles can force that leaving group to leave when they kind of come in and do their nucleophilic attack on an electrophilic center. So all things that we're going to see here as we sort of study how RNase A works. Okay, so let's map on what we knew from our uncatalyzed reaction mechanism. So now we're sort of mapping on our reaction arrows, and we'll kind of highlight what's happening here. We're going to have a 2' hydroxyl, and this is what makes DNA and RNA different, right? This is what's going to allow for this chemistry to happen with RNA and not with DNA. But we're going to have nucleophilic attack by this 2' hydroxyl on the phosphorus. Okay, that phosphorus is electrophilic. It's almost like a carbonyl. We can actually have, there's actually debate in the literature whether or not we have sort of the formation of what we call a pentavalent intermediate, which decomposes and then has the leaving group leave, or there's some research out there to suggest you might have nucleophilic attack and then direct departure. Not important for what we're going to look at here, because in either case, we can make this nucleophile more nucleophilic by having a base deprotonate it, and we can make this bond more susceptible for breaking and this leaving group leaving if we protonate that leaving group. Okay, so what we're seeing here is we're seeing protonation and deprotonation events that are going to happen in our uncatalyzed reaction, and then we'll map on what we see then when we see our enzyme active site. So walking through this chemistry again, nucleophilic attack on our phosphorus here of our phosphodiester bond. Ultimately, we have breaking of our phosphodiester bond, and then we have our downstream nucleic acid piece here. We end up with what we call a 2', 3' cyclic phosphate intermediate. And what we're going to see now is the involvement of water. So water is going to come in here.
I didn't get that.
Water is going to come in, nucleophilically attack this phosphorus, and that's going to allow for the breakage of this bond. Once again, if we have something that we want to act as a nucleophile, it can be made more nucleophilic by a base deprotonating it. If we want to have something be a good leaving group, we want to break a bond, we can facilitate that by protonating that. So at the end, we end up with our upstream nucleic acid and our downstream nucleic acid. Now, truthfully, we did that chemistry in this first step, but I want to map some things on here. Take a look at this guy. This guy in this position right here was going to be a good acid catalyst. It was going to be donating a proton. What if this same guy was right here and then acted as a base catalyst? So protonating in step 1, deprotonating in step 2. That would be important to getting it back to its proper identity, protonated state. Okay, same with this guy up here. Look up here. This guy is going to be a base in sort of step 1. And if it's the same guy, it could be an acid in step 2. So that's sort of what we're starting to see here, is that sometimes we have 2 steps happen because that allows our amino acid residue, if these are amino acid residues here, to get back to their proper states. Okay, so mapping on, again, what I just said here, what if this base was what we call a general base? General meaning we're not identifying necessarily what's happening here. But as we'll see, there's going to be histidine residues in RNase A that are going to act as a general base. Here we've got a general acid, and then those roles flip once we get to the second step. Now we need a general base here and a general acid here, and that's exactly what we're going to see in our enzyme active site. So remembering 3 steps that we always need to see happening with all enzyme chemistries. Chemistry on the substrate, use all your reagents, which generally means water, and then return the enzyme to the enzyme active site to its active state, which means we have to make sure that everybody's in the right protonation state, nobody's still connected to anybody that they're not supposed to be, and that's going to be the 3 steps that we're always going to see. Highlighting with all of the enzymes we're going to study, that first step, chemistry on the substrate, happens in Act 1. Act 2 is really about water coming in to make sure that it gets used, but also regenerate the enzyme active site into its active state. Okay, so let's see if we can see that here. Chemistry on the substrate. Chemistry on the substrate is breaking that phosphodiester bond, breaking this bond right here. Check. We're done with that after step 1. Step 2 is going to have water come in and do its chemistry, and importantly, regenerating those amino acid residues in their active states. So if we had a residue here that acted as an acid, by it acting as a base in step 2, it's regenerated in its active state. If we had a residue here that was acting as a base, by acting as an acid in this next step, it is regenerated. So we can see all of these 3 steps are involved in RNase A here. All right, are you ready? Let's put this together. Now remember, your enzyme speak might be a little bit different. You don't have to have the exact same words, but the more that this just becomes familiar, the easier it is to kind of apply it when you have a situation you haven't seen. So remember, when you're starting this, always look where your arrows start and then start with naming that residue and what it's doing. So in this case, we're going to say histidine 12 acting as a general base catalyst. Notice it's deprotonating there. Serves to deprotonate the 2' hydroxyl of the substrate RNA, increasing its nucleophilicity for attack of the electrophilic phosphorus on the phosphodiester bond. Now that was a long sentence, so don't hesitate to put a period in, take a breath, and make a new sentence, especially if there's more than one residue that's participating. So now what happens? Next set of arrows, cleavage of the phosphodiester bond is facilitated by histidine 119, acting as a general acid catalyst to protonate the 5' hydroxyl of the downstream RNA, whose departure, so once we have this guy, departure is going to allow water to then diffuse into the enzyme active site. All right, end Act 1, seen, right? Think about what we've done. We've done chemistry in the substrate. We broke this phosphodiester bond, but we haven't yet involved water. It's coming onto the scene, and we haven't regenerated our enzyme active site. Look at what we look like here. This histidine 12, to be active, needs to be deprotonated. This histidine, 119, needs to be protonated. Excuse me. Look what they look like over here. They're not the right protonation states. This histidine 12 is now protonated. This histidine 119 is now deprotonated. But water has come in, and we still have another step that's going to allow us to regenerate that enzyme active site. Okay, so next stage. Start where your arrows start. They start down here this time. Histidine 119, now acting as a general base catalyst, serves to deprotonate water, increasing its nucleophilicity for attack on the electrophilic phosphorus of the 2', 3' cyclic intermediate. Opening of this cyclic intermediate, and that's breaking that bond, okay, is facilitated by histidine 12, now acting as a general acid catalyst to protonate the leaving group 2' hydroxyl group. Reaction on the substrate is now complete. All reactants have been utilized. That's step 2. And now the third step, the enzyme active site has been regenerated. Okay, so this one's pretty straightforward. We can see now we have our upstream nucleotide has now sort of departed. And if we look back at our enzyme active site, it's ready to do additional chemistry. Histidine 12 is in its deprotonated state. Histidine 119 is in its protonated state. And when we role played this and acted it out in class, right, those of you who were the histidine residues, you had a little name tag that said, my native state is deprotonated, protonated, depending on what you needed to do to initiate that initial step of the reaction. All right, so that wraps up our understanding of RNase A.